Great, fantastic. Thank you so much for the invitation to be here and tell you a little bit about what we're doing. What I'm hoping to do in the next 30 minutes or so is to convince you, hopefully it's not too difficult to do, but to convince you that by combining microfluidics with electronics and the right biological applications, that there's a lot of potential there. I'm going to give you two stories that serve to illustrate that point. One that involves cells and one that involves molecules. And I want to start by talking about the biology or the physiology, and in particular, the immune system. So we heard this morning a number of talks that touched on the immune system, and that's what I'm going to focus on here. Here's a plot of the top 10 causes of death, and you can see that diseases that touch the immune system are here, so infectious diseases, obviously, but also autoimmune diseases like diabetes are there as well. But even things that don't obviously involve the immune system, like ischemic heart disease, in fact, when you look at the mechanism for how they act, well, there is the immune system. So here's a little cartoon for the physiology, and you can see macrophages here, which are a cell of the immune system. So that goes to the point that the immune system is involved across a lot of different indications, even things like cancer. So this is just a little cartoon that shows the CAR T therapy and more generally immuno-oncology, so harnessing our immune system to fight our own cancer. So again, here's oncology, but it involves the immune system. So I'm an electrical engineer, and one of the things that we look for are things that are platform-type technologies. And I'd like to argue that the immune system is really a platform for physiology. So if we can build tools that help us monitor the immune system, we can use it across a lot of different indications. That gives us a large market, right? And that makes it worth it to integrate microelectronics in there. I'm going to focus in on one particular disease here, which is sepsis. So sepsis is a whole body response to an infection. So you have an infection, it maybe gets into the bloodstream, and basically your immune system overreacts. So even though you might go and kill, let's say it's a bacteria, you go kill the bug, that does not make you healthy, right? It's the immune system overreaction, the dysregulation, that is what causes the morbidity and mortality associated with sepsis. And so the clinicians have to somehow get your immune system, which has become completely dysregulated, back into a regulated state, and they have to do that quickly. So sepsis has a large burden in terms of dollars, in terms of people, and importantly, sepsis is something that occurs very, very quickly. So we're talking about hours type time frame, where you can go from beginning to very, very sick. And when you're if you have sepsis, you go into the critical care unit, and you're going to be hooked up to monitors. They're going to monitor your blood pressure, your heart rate, your SpO2, all these types of things. But what they're not monitoring directly is the immune system itself that is this central player in the pathophysiology of sepsis, right? So how are we monitoring the immune system? Well, we're going to be doing that, for example, with once or twice a day blood tests. So the CBC, complete blood count, is a very common one that's ordered, right? And so if you're talking about a disease where the dynamics are hours and you're measuring things where the dynamics are 12 or 24 hours, you're not matching it appropriately, right? And in fact, when you're doing your CBC, you're taking a census. You're saying, here are all these types of cells, here are all these types of cells. But the cells are doing something. And something that our clinical collaborators came to us and wanted to know is, can I not just know who in the immune system is there, but can you tell me what they're doing? Can you give me some information about their function that would help me improve the care that I deliver, right? And so that led us to, well, are there ways that we can develop that we can quickly and directly assess the immune system function? And that's what I'm going to tell you about here. So we're going to talk about cells, we're going to talk about blood, and I just thought, let's figure out what's actually in the blood. So our blood is about half water, or liquid. That's our plasma. That's where we're going to measure molecules. That'll be in my second part. And then the other part are the cells. Almost all of those cells are red blood cells. But the cells that we care about here are the white blood cells, the leukocytes as they're called, and in particular the subset of leukocytes called the neutrophils. These are the most abundant white blood cells in humans, but it's still a tiny fraction of the blood, okay? So those neutrophils are normally, they're sort of going around in your circulation. They're in a resting state. And when they see molecular signals that are coming from some place where there's inflammation, they become activated. And they undergo a number of different actions when that happens that are shown here on the right. But what's interesting is that the neutrophils themselves are acting as little computers. They're taking in molecular signals, they're computing on them, and they're making a decision. And one of those decisions is, should I stay resting or should I become activated? And so we wanted to look at that decision and monitor, are the neutrophils in your blood activated or resting? This is an assessment of leukocyte function, of neutrophil function. And can we do that easily and quickly? So now I'm an electrical engineer, so, well, I look at it as an equivalent circuit. And it turns out that neutrophils can be modeled as a bunch of resistors and capacitors. And we want to detect changes in that. This is kind of the same as, well, when you have a touch screen, the touch screen itself is detecting the difference between air and your finger, right? And the difference it's detecting are that those have different electrical properties, the conductivity or the permittivity of those materials. Well, cells are also made up of materials, and they have conductivities and permittivities, and we can detect differences there. And the question, or the key challenge, is if there are differences that correlate with the state of the cell, well, then maybe I can turn that biological information into electrical information, and now I can use microelectronics in order to read that out, okay? And that's what we've been working on. So the fact that cells have electrical properties is not new. That goes back to Maxwell. But can you actually detect those properties in a sensitive and specific way? That is kind of hard. The way that we're going to do it is to use a technology called dielectrophoresis. So I need to have a couple of slides of physics here. Dielectrophoresis is the force that occurs on an object. It doesn't matter what the object is. Any object that's different than free space, when you put it in an electric field that is not uniform in space. And that's what's shown in this little cartoon. The electric field lines here are close together and they're farther apart there, so this is not uniform in space. And in this situation here, the particle is less conductive, if you will, than the medium that it's in, which might be water. And in that case, it'll get pushed to the bottom. If the particle was more conductive, it would get pushed to the top. So there might be a situation that's right in the middle where there is no force at all, and that would be if the particle is what's known as electrically transparent. This is the exact same thing, really, as if something has the same index of refraction as water, and you put it in a glass of water and you can't see it anymore. It's optically transparent. Here we're looking for a situation where the cells are electrically transparent, okay? And so what we do is we create a gradient of electrical properties, and that's actually really easy to do in liquid. You just change the salt concentration. So you have salty liquid on one side and not salty liquid on the other side, and then you make a gradient. If you put particles in there and apply this DEP force, then what happens is they'll line up in a place that tells you about their electrical properties, okay? What's interesting about that, or at least what's shown in this cartoon, is that that is independent of the size of the cells. And this is very important because we have lots and lots of microfluidic techniques out there that can move cells around, and almost all of them are dependent on the size of the cells. But any reasonable cell population has about a factor of 2 variation in size. So if you don't do it right, all you're going to end up doing is separating big cells from little cells. And that's not what we want. We want to separate activated cells from unactivated cells, and we don't care how big they are. Okay? So we need an approach that is insensitive to the size, and that's what we do here. So how does this work? This is called isodielectric separation, and what it ends up being is a little chip, which is about 1 centimeter by 3 centimeters. Sorry, 1 inch by 3 inch, 2.5 by 7.5 centimeters. And we have liquid that goes through it, so it's a little microfluidic device. That liquid has a gradient of conductivity, which is shown by this color scale here. And as the cells enter in, they encounter these electrodes that have a small electric field applied to them, a little voltage, and that separates the cells. And that's what's shown here in this little video. And that separation of these two different colored objects depends on the voltage and the frequency that you apply to this system. And so what we do is we take our cells, we separate them, and we get their location in the channel. We get these histograms, and that's the information that comes out of this type of system. Okay? That's what we call the IDP, the isodielectric point of each cell, and that's what we're going to do. We've done this now for about a decade and a half with all sorts of different types of populations of cells. The one I'm going to focus in on here are neutrophils, so a type of white blood cell. So we did a lot of work to show that this worked in cell culture and that it worked in animal models. I'm not going to talk about that at all. I'm going to skip that and focus here on the clinical work that we've been doing. And so we worked with colleagues at one of the hospitals in the Boston area, Brigham and Women's Hospital, and they have sepsis patients that they see in the ICU. And what we do is we take 50 microliters of blood from them. So this is a pretty small amount of blood. And that's actually really important because even though these patients may have a central line, so they can actually take blood whenever they want, if you take too much blood too often, you can actually hurt even adult patients, right? So actually small volumes of blood is important even in adults. So we're going to take this blood and we're going to separate out the neutrophils, so this most abundant type of white blood cell, and we're going to put them through this IDS device. And the goal is that this will separate them into activated and unactivated states. So this is what it looks like from a different point of view. So it's a little microfluidic device that has a channel here, a certain width and a height, and then it has these electrodes that apply an electric field that are going to, when the cells encounter them, they will get separated according to their properties. And the idea being that cells that are activated and cells that are not activated end up in different locations. So then we get a histogram of those different populations, and we can then assess what's going on with that patient sample. One of the things that's important in how we do this is that we have an internal control. So we want to understand the difference between activated and unactivated cells. And one of the things that we do in order to do that is that we take that sample and split it and then activate all the cells in there. And we use a chemical called PMA. We've used other chemicals since then, but this is an early one. And it basically turns the cells up to 11, if you will, in terms of their activation state. And then we can compare that with what we call either the vehicle or non-activated state. So this is blah, blah. This is what it looks like. So these are just images of cells going through a channel from a patient here. And these are the cells that are just from the patient. And then we have generate a histogram that's shown there. And then we have cells from that patient where they've also been exposed to this PMA molecule that activates them as much as is humanly possible. And then we generate that histogram there, okay? And we're going to do that over and over for different patients. The very first thing that you have to do is to figure out what are the conditions that properly discriminate these cells. And that's work that we've done, and that's what's shown here, where we look at the position in the channel, this median IDP, as we vary the electrical frequency that we apply to the electrodes. And what we see is that if you go down to about 7 megahertz here, there's a nice distinguishing between the blue and the red curves, or points, which is typically the frequency that we operate at. And we found that this frequency is the same whether we're using animal models, human patient samples. So we've actually stuck with 7 megahertz for a very long time. Okay, so this is what the data looks like coming out. These are samples from healthy individuals, 2 different samples, 2 different individuals. Okay, and we have 2 sets of curves. We have the cells that come out of the device called vehicle. That's just the regular cells, and the cells that have been chemically activated, and that's called the PMA. And you can see that the red ones are generally to the left of the blue ones, and that's the same for both of these samples. If you look, meanwhile, at samples from a septic patient, well, you see that everything is shifted to the left. The blue has shifted to the left, and the red has shifted to the left. So both the vehicle as well as the PMA-activated samples. And then we look at a number of different summary statistics from this. So one is we can look at the medians of the populations. So that's these dotted lines for the blue and for the red. And if we plot that, we can see that in the vehicle situation, we can tell the difference between patient samples that come from healthy individuals versus samples that come from individuals with sepsis. So that's good. But what's interesting is that if you look at what we call the delta median, meaning the difference between these two, we actually get even better performance if you look at the difference now between healthy and septic patient samples. So this is pretty cool. We can tell the difference between healthy individuals and septic patient samples, so that's good. That does not mean that we would ever use this to diagnose sepsis. This is a terrible way to diagnose sepsis because it is very sensitive, but it is not very specific, meaning that there's lots of things that will alter your immune system. You could have a cold. In fact, when we buy healthy blood samples in the Boston area in the winter, they are not all healthy, right? People will donate blood for medical testing, and they'll say that I'm healthy, and then we'll get them and we'll analyze them and find out that no, this person is actually sick, right? So I'm just saying that the immune system lights up for a number of different reasons. That does not mean you want to use this to diagnose them, but it does mean that if you know they have sepsis and they're in your critical care unit, that you could use this potentially to monitor them as their sepsis evolves. That's what we're trying to do here. I just want to be very clear about that. Okay. So what we did then is we looked at this metric, this delta median IDP from this microfluidic device, and compared it to a bunch of existing things that clinicians would use. So one I mentioned very early was the CBC, the complete blood count. And this is the total number of neutrophils in the sample as a function of this thing called the SOFA score. The SOFA score is a pretty well-established clinical score that goes from 0, meaning healthy, to I think the max is maybe 24 or so, which is basically dead, okay? And what you see is a pretty poor correlation between neutrophil counts and your SOFA score. And in fact, we looked at a bunch of different markers that people use, and the delta median IDP was the single most highly correlated marker amongst anything that we looked at. We see things like, for example, if you are on mechanical ventilation, you are probably sicker than if you are not on mechanical ventilation. For those of you who remember COVID, we remember that people would get this thing called ARDS, and the very sickest people would go on mechanical ventilators, right? So that tends to be correlated with sickness, and in fact, your delta median score is lower when you are on mechanical ventilation, which is consistent with having a higher SOFA score, which is also sicker. We would also see things like recovery. So here's your white blood cell differential over time, so healthy individual, and then day 0, so you typically when you're at your sickest and as you're recovering over time, and you see there's really no correlation. Whereas when you look at the PMN function that we're getting here, you can see that you start to recover toward healthy as time goes on. So this makes us pretty excited, but is still not there. So what is some of the technological things that we have to do? One is to decrease that blood volume even further. So we're at 50 microliters. That seems like not a lot of volume, but it's still more than you can get from a finger stick. So you have to do phlebotomy or get things from a central line. It turns out that one of the things that we've learned in doing this is that getting your device, your system into the clinical workflow, you have to accommodate what they do, and they're not going to accommodate what you want, okay? And so being able to take finger sticks is a lot easier for the care staff than getting, let's say, even 1 ml of blood multiple times a day. And so we've been working toward that. We have a way of doing this. So typically the way that you take the whole blood and you get the neutrophils out is you do either things like immunomagnetic separation or density gradient. You can't do that at small volumes. We've used an inertial microfluidic approach to do this. This is with the Hahn lab at MIT. And basically it's like going around a curve very, very quickly. You take the cells, you pass them through this little spiral very, very fast at high throughput, and it pushes all the cells to the side like you would in a car if you were going around a curve. And then the second part separates the larger cells from the smaller cells, and the larger cells here are the neutrophils. And so this is a very fast way of getting the neutrophils out of blood. And we've worked to decrease this to be able to work with 10 microliter samples. So maybe I won't talk about all that, but we can do it. Here's data showing from finger stick samples that we can do things. Turns out this is also interesting for people who work with mouse models, because mice have about a 1 milliliter blood volume. So if you can do something with 10 microliters, that's much better than if you can do it with, let's say, 100 microliters. So what I think is the opportunity here, right, is to be able to take that immune system and monitor it very, very frequently by doing things with just a finger stick instead of with phlebotomy. And there's a whole range of situations where you might want to do this. I talked about sepsis, but we've talked with folks who are interested in things monitoring people post-transplant. We talked about CAR T therapy, ARDS, as well as at home. So we heard from some of the startups trying to bring things to the home. Well, maybe you could monitor at-risk populations so that they wouldn't be readmitted or be admitted earlier. On the other hand, we have some technological opportunities, right? Which is, can you take everything and now really fully integrate it? So instead of having kind of a distributed system across a few different components, can you put everything together into an integrated fluidic electronic system and do everything all at once? The second thing I want to do in my last few minutes here is talk about molecules. So the first one was measuring cells by using electronics and microfluidics. Let's talk about molecules, because molecules is the other half of this. Proteins are an interesting molecule because, for the most part, I'm going to say something which is not true, but almost true. All the protein assays are measured the same way, which is using an immunoassay. So if you can develop a technology that can perform an immunoassay, again, it's a platform. You can use it across a number of different proteins that you want to measure. I'm going to talk here about cytokines. Cytokines are one type of protein, and they're important, again, across a number of different indications. So here's what's known as cytokine release syndrome or cytokine storm following CAR T therapy that you want to monitor a few days after the therapy. But also we've talked with people that are interested in autoimmune diseases. So chronic autoimmune diseases like multiple sclerosis, where you have a years-long progression and you have flare-ups. Could you start to see that flare-up at home earlier on, adjust your therapy, and minimize the flare-up, things like this, okay? So protein measurement's another platform that we can apply microelectronics to. Of course, there's many companies that want to do this, that already do this. All of us have had now experience with this one down here at the bottom, COVID rapid testing. The ability to get an answer as to whether I can go visit grandma today or I have to wait a few days, but getting that answer within 15 minutes is really enabling. You remember at the beginning of the pandemic, we would have to get the RNA tests. It would take a couple days or a week, and you would just not know what could go. Once you get things in 15 minutes, it's much better, right? So fast is important. But that's just a yes-no answer. There's a lot of situations where we want to get quantitative information, where we want a lower limit of detection, LOD, where we want to get multiple pieces of information, so we want plexing, where we want low cost, okay? And that's what we're focused on here. So we have a system which goes from blood all the way to an answer in about 30 minutes. It first does sample preparation. I'll actually talk about that last. And then it does basically an immunoassay on a little microfluidic device. And it includes microfluidics and electronics. And the reason we do that, right, is because the microfluidics is good for control and manipulating of volumes that are very, very small, like finger stick or smaller, right? So 1 microliter is a cubic millimeter, and that's often plenty of liquid to do your measurements. But what's interesting is that we think and we believe that the sooner you can turn your molecular information into electrons and bits, the better. And so we tend to prefer a direct electrical readout using electrochemistry over something that would be an optical readout that you would then convert into electrons, okay? What's interesting is that in our lab, we have rack-mounted electrochemical front ends, electrochemistry front ends that cost thousands of dollars. And then nowadays we can go to TI or analog devices and buy for $5 or $10 a chip that'll basically do the same thing, right? So the kinds of measurements that you can do nowadays are just amazing. We just have to bring those together into complete devices. We're going to use what's known as a bead-based assay because it'll give us a little bit of amplification. Okay, so how this works is we have a little microfluidic device, and inside we have little beads. And on those little beads, we have these things called antibodies. Those antibodies will bind to the molecule that I care about, the protein. Also on those beads are in these little, whatever that shape is, enzymes, okay? So we first bind the molecule onto the bead, and then the molecule binds onto an electrode here called the working electrode, which is part of our electrochemistry system. Okay? And then we bring in a reagent, which then the enzyme acts on and it generates charge. And we're very good with electronics at counting charge, right? So counting single electrons, actually not that hard. So that's how we do it. That's how we turn the abundance of a molecule into electrons, okay? So we have a little microfluidic device. It has little valves and all these cool stuff on it. Let me just show you a little bit about how it works. But initially, you bring in the sample into this first part here. There's a little on-chip pump that pumps the sample around and incubates it with the beads. And that allows the analyte to be captured on the beads. Then we switch things up and move everything over here where we have a magnet that pulls down the beads. The beads are also magnetic. And then over here, we then mix it so that the beads can bind to the electrode, okay? And then we add in our substrate at the end, and we just measure current, okay? So that's how it works. Here's an 8-channel device with electrodes on the bottom, microfluidics on the top, valves, all sorts of fun things. And here's the associated electronics, here with an analog devices front end to do all the electronics, the valving and whatnot. And I'll just sort of show you a little bit of data. But basically, we can get the same results as you can with the conventional approach, which is called an ELISA. It's an enzyme-linked immunosorbent assay, basically how you would run it in a central facility. Similar limited detection. But the difference is that this is 40 minutes, so it's much faster than the other one, and it's all automated, okay? We've also integrated the sample prep. So here we are using a microneedle device from this company called YourBio, and it draws blood painlessly, and we can take that blood then from this device and then bring it onto our device and use a little membrane filter to get rid of the red cells so that we just have, or get rid of all the cells, so we just have the plasma. So I just won't go through that. So again, we have a device which combines microfluidics and electronics to monitor the immune system. Again, there's a whole number of different applications, but I think here you can actually start to push even further, not with a bead-based assay, but with different types of immunoassays to not just do portable, but think about actually wearable. And that, I think, just like continuous glucose monitoring versus the finger stick blood glucose monitor opens up different applications, so with this here. On the flip side, I think there's pretty interesting technological opportunities in terms of thinking about the integration of the microfluidics and the electronics, making everything smaller to lower the cost, things like that. So what I wanted to tell you about is microfluidics, electronics, biology. I think there's a huge opportunity there. I think Taiwan has actually an important role to play here given the ecosystem that exists. I think there's a wide range of use cases across from inpatient to at the home to on the patient. But I think it's not trivial. If it was trivial, we would have already been doing it, right? So I think one of the big challenges is that microfluidics, even though it is micro, they're still pretty big, right? And if you were to calculate if you were to make your silicon that size, it would be exorbitant cost. So silicon we want to make as tiny as possible, the microfluidics can only be so small because you need to, to manipulate a microliter, let's say, of liquid, right? And so figuring out how to do what we would call either heterogeneous integration or advanced packaging, but I'll argue it's like super advanced packaging because it's not just electronics that you're trying to package. You have to package fluids and molecules and all these things. And then I think the other challenge is that as we measure more things, as we measure things more often, or measure new things, you have to somehow scaffold that onto existing clinical information, right? Because that's what the doctors, the physicians know about. And if you come in with your weird widget that measures things in a way that no one knows, they don't know what to do with it, right? So I think that's another challenge, but we're up for it. Okay, thank you. Oh.